Hello and welcome to this BioProcess International Ask the Expert webcast. I'm your host, Leah Rosen, the online editor for BioProcess International. Before we get started, just a couple of notes. This webcast is being recorded and will be made available for replay in the multimedia section of our website. We've muted the audio lines, but we welcome you to type in your questions for our speaker in the question and answer window on your screen. After the presentation, we will begin the question and answer portion, and I will ask our speaker your questions. Your questions in the question and answer window will only be visible to myself and our speaker. So thank you for joining us today. It is now my pleasure to introduce our speaker, David Cetlin from Cygnus Technologies.
Thank you so much, Leah, and thank you all for taking time out of your busy days to attend this webinar. I'm happy to deliver this message about a new and innovative way and approach to viral clearance. So my talk is titled Predicting Viral Clearance, Design of Experiment, High Throughput Screening, and AAV Case Studies utilizing a non-infectious MVM surrogate during downstream development. So downstream process development, if you work in the space, you're well aware that it's a complex activity. It requires a lot of resources up front. It's time-consuming, capital-intensive, takes many years to go from screening different resins and techniques, filtration devices, to a validated process. During the course of those years, we as an industry have a very nice toolbox which gives us the ability to look at process impurities such as host cell protein, host cell DNA, residual protein A, benzonase, and really that gives us a lot of data during the course of process development characterization and helps the evolving process into something that can be accurately validated and perform well for you at scale. The one thing missing from this toolbox is any tool that can give you a glimpse into viral clearance validation, which occurs late in that biopharmaceutical lifetime. The reason why there is no viral clearance kit on the market, or there hadn't been until Cygnus had introduced that, is because of the nature of viral clearance studies. Viral clearance involves BSL-2 or sometimes BSL-3 infectious agents. And because of these heightened sensitivity on these viruses, typically experiments are conducted at CROs. Because of the logistics of working with the CRO and the expense, most companies delay these types of tests until very late into process characterization, or maybe perhaps just prior to process validation. And from experience, I know that that is a poor time to find out that perhaps your Anion Exchange column isn't giving you the full range of clearance that you were expecting, or let's say the virus filter begins to clog up on you and you have to retool that ideal process step, maybe look at new technologies. I know from experience that that was the case when I was in the field, and so it was something that I wanted to address, and that's where this idea of the MockV MVM kit sort of came from. So what we're doing here is we're replacing the live viruses used by viral clearance validation labs with what we call a mock virus particle, or MVP. And the idea is to have different MVP kits that address the different viruses used internationally by CROs. And the way that a study will work is by first spiking your process load with this mock virus particle, and then processing that spike load through your downstream process step, whether that's a chromatography column or a virus filter. So on the other end of this step, you have your flow through, your wash fraction, or your elution pool, and now you can quantify the amount of particle that had been there relative to the amount that now you've collected in these samples. From that, you can derive an LRV, a log reduction value. So in essence, it's very similar to a live viral clearance validation study. The only difference is we've replaced that live virus with a mock virus particle. So what's paramount in this approach is how well this mock virus particle is in fact mimicking the viruses that are used internationally for validation studies. The way that we sought our first candidate was to attempt to form a particle that mimicked MVM. MVM is an internationally used parvovirus for pre-IND or pre-BLA type of studies. This, along with XMULV, are the two marquee viruses when it comes to viral clearance validation. So the way that we've constructed our mock MVM particle is we took the major capsid protein, the VP2 capsid, and we express that protein recombinantly in a baculovirus SF9 system. So these SF9 cells are cranking out the VP2 protein. They self-assemble into what you find here on this image on the bottom right of your screen. That's a TEM image of the particles that we can produce. So as you can see, this is a very purified, very nicely purified prep. The concentration of these particles typically are in excess of 10 to the 12 particles per mil. So it's a highly pure, highly concentrated stock of these particles, which we hope will mimic MVM. And so to that end, the first study that we did was we collaborated with the FDA. This was with Kurt Borson's lab at the FDA, and we asked the question, how well do our particles really mimic the physiochemical properties of live MVM? And so through his lab, we were able to screen the 2 particle types along with PP7 bacteriophage. So you see on the bottom table here, we have 4 columns. One shows you the analytical technique that we tried, and then the 3 columns on the right are the different particle types, starting with live MVM, our particle, the MVP, and the PP7 bacteriophage. So you can go down by each row, analysis by analysis. It looks like our particle is stacking up very nicely against MVM, where PP7 does a good job of mimicking the size properties of MVM, but when you get into the surface charge, surface hydrophobicity, that's where that particle sort of lacks, and our particle is able to provide a better surface chemistry, better surface surrogate. And again, we're not surprised at this result. We did use MVM's natural capsid protein in the construction of this particle, but it was very nice to have that proven out during the study. So with that data, we marched forward thinking we have a very nice, accurate surrogate for size-based or charge-based separation. And the only question remained was, how can we quantify these particles? They're not infectious, and they do not contain nucleic acid, so we can't do a TCID50 plaque assay, nor can we do a qPCR-based assay. So after attempting a simple ELISA, we migrated towards this immuno-qPCR approach. The reason why we chose this over an ELISA was we were able to expand the dynamic range of the assay. So now we're able to deliver 4, sometimes even 5 logs of clearance when this assay is used for a spiking study. The next couple slides, actually the rest of the slides I have, will just show you applications of our kit, of this technique. The first application was within the gene therapy space. The purpose of this study was to assess the ability of MVM clearance through the use of our kit in a typical AAV process. This was a HEC-293 platform. We collaborated with Regenex Bio for this study. So the unit of operation that we were looking at during the study was an affinity column. This is porous AAVX. It's a Thermo Fisher resin. So columns were provided to us, and we operated our steps at either center point conditions or worst case. And the loads for either of these runs were spiked to a starting concentration of 10 to the 10 MVP particles per mil, or with MVM according to TechCell standard procedures. TechCell was the CRO that we used for the MVM portion of the study. So after performing all of the spiking runs at either center point or worst case conditions, we stored samples and analyzed the MVP samples via the immuno-qPCR assay or the MVM particles with the plaque TCID50 based assay. So the first set of results that we'll see are from our particle, the MVP. What you see with the first two center point runs is with the elution fractions, we see very little MVP. This means that the AAVX resin was able to retain our particle while allowing the AAV, which is what it's supposed to have affinity for, to come through. Or sorry, reverse. It's able to bind the AAV and not allow the particle to come through. That's why we saw very low levels in the elution. And that's why the LRVs are inversely very high. Indeed, where our particle did come out is during the flow-through predominantly, where you see a 52% percentage there in center point run 1, and a 67% of the total particle challenge coming out in the flow-through for center point 2. The other fraction where we saw our particle elute out was during the benzenase wash step. So by the time elution occurs, there's very little particle that remains bound on that column, which is why we see these nice LRVs. Those were both with center point. When we changed the conditions to load a higher capacity onto the resin and slowed the flow rate down to, which is what was thought to be worst case, we did see an impact in terms of the LRV. From about an LRV of 5, we now have an LRV of about 4. So we lost 1 log of clearance by changing those parameters. Now, how did it work with MVM? This was again done at TechCell, and what we saw here was very similar. You see the flow-through fraction at center point had 55% of the particle break through. Interestingly, the benzonated wash step didn't have that 10%, but nonetheless, when you get back down to elution, you're eluting off very little, and we had robust effective clearance of MVM, 4.35 logs. That was at center point. When these experimental conditions were changed again to worst case, high resin load and residence time, we saw again another drop in the LRV, about 1 log. So the story was about the same for both particle types. The next slide summarizes that nicely in this graphic. On the left-hand side in blue, you have the MVM LRV results for center point, and in the red, you see the MVP LRV results for center point. Then on the right-hand side, the same style of reporting the results, but now with the worst case parameters. So you see, likewise, these drops in both particle types when you shift from center point to worst case. So this indicated that our approach to viral clearance with this non-infectious particle could indeed predict the outcomes of the MVM studies. So we're currently drafting a manuscript which will detail that study in much greater detail. The next study was an Anion Exchange study that we did in collaboration with GlaxoSmithKline. I won't spend too much time on this. This has been published in BPI. If you're looking for that article, please reach out to me after the presentation. I'll be happy to send you a PDF. So what GSK did was they designed a full factorial study. They looked at pH and conductivity and the impacts that those two parameters have on Anion Exchange's ability to retain virus, and the virus, of course, being MVM. So the first 6 runs, runs 1 through 6, were done with MVP and MVM in parallel, and so that's what we're seeing on this slide. And then the final 4 runs were conducted just with MVP alone. So let's turn our attention to the first 2 runs. They're a good way to highlight the data. The first run was conducted at a pH of 7, conductivity of 3, and what we see is a nice effective clearance of MVP, 4.17, second run 4.46, and when we performed this at TechCell with MVM, we likewise saw robust clearance at 4.11. So this indicates, although your pH is low, which is typically not desirable, as long as you keep the conductivity at a minimum, you can expect good clearance. However, when we shift that conductivity now in run 2 to 13 millisiemen per centimeter, you see this dramatic drop in the LRV results. So we go from 4 roughly to 2 in both the MVP and the MVM. So what GSK did with this data, they took all the MVP data, not the MVM, just the MVP, and they plugged that into their JUMP software, and they were able to look at how the 2 parameters impacted each other, and they were able to generate this, this heat map that showed the optimal space to operate their process. So in conclusion, we found through this collaboration that MVP did indeed serve as an accurate indicator of MVM removal. GSK was able to statistically build a valid model from this data set. We built this model with just the MVP data, so we did not require any of the MVM. And again, we published all of this data in a journal, a BPI journal. So for a complete look at what we did and what we accomplished here, I please refer you to that journal. Thank you. Okay, last but not least, we conducted a high throughput screening study with the NIH. The NIH utilizes their TCAN system to scout new resins, new separation techniques. But one thing that the TCAN cannot do is travel well. You cannot bring your TCAN into a CRO. So it's very hard to conduct these sort of high throughput screening studies and generate any meaningful viral clearance data. So that's where our non-infectious technique comes in. What they did was they used robocolumns, 8 different resins they screened at 3 different pHs apiece, and they looked at the ability of these resins at these different pHs to retain our particle as higher and higher and higher conductivity buffers were applied to these columns. So the results kind of speak for themselves. The amount of work that you can do in this high throughput screening mode. So the first column on the left, you see the different resins and at the different pHs. They're labeled 1 through 8. And then the next column to the right shows the flow through, the ability of these resins to retain the particle. However, as you increase the salt conductivity that's applied to these columns, there are differences that occur in their retention of the particle. So for example, resin 3 had great retention of our particle even up to 300 millimolar salt, same with resin 8. Whereas other resins such as 1, 2, maybe 6, 7, they lost the ability to retain that particle as they were exposed to higher saltier conditions. So the data from this study was also repeated with a Anion Exchange battery of resins, and we also tested some membrane absorbers as well. The data from this study is being submitted for publication, and we're excited for that to come out because we'll reveal what these different resins were when that data comes out. So last two slides, just to sum it up. We commercialized this kit, made it available to the industry for your uses, whether it's DOE, high throughput screening within the gene therapy space, what have you. And what it is it's a kit that comes with the spiking elements. So it gives you the ability to perform your own BSL-1 spiking experiments, and it gives you the quantification components to conduct your own assays. So, you know, it's like host cell protein kit, but now you can extend that sort of mind state into the viral clearance mode of impurity removal. And where we see our customers utilize this kit is really throughout the lifetime of their activities. Early on, you can screen new resins that hit the market. Let's say, you know, there's a promising technique, but you don't want to pay all the money at a CRO to get the viral clearance data, and so therefore you kind of just drop it from your radar. Well, this kit, you know, can give you like the ability to de-risk that decision to like maybe try it out. And let's pack a column, let's do a DOE, let's actually play with this new resin and not worry as much if you were able to vet that with our economic BSL-1 kit. Places 2 and 5 are before conducting a live viral clearance study. You might want to generate some preliminary data with our kit to expose any potential issues. That would be like an insurance policy before going to a CRO and spending the big money. And then the sweet spot where we see most of our customers apply the kit is 3 and 4. This is when process development and process characterization slash optimization activities are really picking up. So on that note, I'll end the slide deck and open up the floor for any questions that you may have.
Okay, great. Thanks, David.
Sure.
So the first question is, how many experiments can I perform with the MockV MVM kit?
That's a good question. And the answer to that really depends on the volume of your load and the concentration that you want to spike to. So the assumptions that we built into the kit were that loads would be 150 mils, and we'd provide you with enough particle to spike 150 mils 10 times at a concentration of 10 to the 9 particles per mil. So just to reiterate that, if your load concentration is going to be spiked to 10 to the 9 particles per mil, and you have 150 mils of that spiked load, you can run 10 experiments in that mode. We actually sell the spiking MVP as a solo component in case you have volumes that are higher than 150 mils, or in case you want to challenge at higher levels, like 10 to the 10 particles per mil.
Okay, great. And how does MVP LRV data compare to the MVM spiking studies?
Yeah, so that's the whole holy grail here. And so you saw in the slide deck 3 different studies. 2 of those had comparable MVP versus MVM data built into that. We actually have some other papers published by AstraZeneca and Asahi Kasei, which had the parallel style of reporting as well. We typically see our results come within 1 log of historical MVM data that the customers have, but we maybe 50% of the cases are even better than that within 0.5 logs, but 90% of the time we're within that 1 log range. So it's a fairly accurate assessment. Probably the best use of our kit is, you can use our kit to benchmark MVM, especially with historical data, but the best way to perhaps utilize this is for trending purposes, like what we saw with the GSK data, to map out the conditions and the parameters where you might get poor clearance. That's the best and most proper use of the kit.
Okay, and it looks like we're running short on time, so we have time for just one more question. If we didn't get to your question, we will be passing those to David, so you do have time to still enter your questions, and we'll pass those along as well. The last question is, has the FDA evaluated data obtained with the mock virus particles?
We get this question a lot. So the FDA is certainly aware of this approach. They collaborated with us to generate the physicochemical data, and we've been in front of them in various forums like presentations or roundtables to discuss this as an approach. As of now, this technique is not a substitute for live viral clearance studies. Those are still required for submitting a BLA or an IND. However, with all of the push that our customers are now organically, going after, they want to see this data used in that sort of manner. So when customers bring this up to the FDA, it makes the FDA kind of consider, for places where this data set could supplement, I won't say replace yet, but could definitely supplement live viral clearance data. And with everyone's active use of these kits, maybe we can start convincing the EU, the FDA to look into this even further.
Okay, great. Thanks, David.
Thank you.
And thanks to our audience for joining us. The recorded version of this webcast will be available for on-demand viewing on our website, and as a registered attendee, you'll receive a follow-up email providing with the direct link. We look forward to having you join us at future BioProcess International Ask the Expert webcasts. Look for those announcements in your inbox. Goodbye.